Hi, everyone. So I'm Dr. Ana Moles. I work at the Spanish National Research Council. So I'm group leader of the Tissue Remodeling Fibrosis and Cancer Group at the Spanish National Research Council. And it is my pleasure today to host this session. So thanks to the organizers, Dr. Gracia and Dr. Cubero, for inviting me. And it's with great pleasure that I introduce Professor Prakash Ramachandran from University of Edinburgh. So he's an academic hepatologist and professor of experimental hepatology. He did his PhD under the supervision of John Iredell, Stuart Forbes, and Judith, Klus?
David Klus, who's a nephrologist.
Yeah. And we all know him, so we've worked with his data sets, a lot of us. So I don't think he did further introduction, but he works on the immune cells, the heterogeneity of immune cells, and how they work in liver fibrosis context, both in human and mouse. He received several awards, such as the Emerging Leader of EASL and an MRC Senior Clinical Fellowship. So Prakash, you don't need further introduction. So it is our pleasure to have you here today and looking forward to your talk.
Thank you, Anna. Great. And yeah, thanks to Jordi and Anna for the invitation. It's fantastic to be here. And it's been a great day already. I'm really looking forward to the next couple of days of interactions and discussions. So I had it, when I first sent my title, it was a bit generic, so I'm sorry. So it was immune regulation of liver fibrosis. But actually, what I'm going to talk about today is how we are using single cell and spatial omics technologies to kind of really understand immune cells in the context of fibrosis. And for those of you who are not interested in macrophages, I'm sorry, but you're going to hear more about macrophages over the course of the rest of this session. So I'm going to really use macrophages as an example of how we can kind of start to unpick pathogenic populations and kind of new therapeutic targets using these technologies. So I'm visiting from Scotland. So Scotland, those of you who've been know it's a fantastic place to live, great place to work. And particularly as a hepatologist, it's particularly good because Scotland has one of the highest rates of liver disease in Europe. So those of you who know Scottish people know that they love nothing more than beating the English. And one of the few things they can beat England at, and it's not football, is the rates of liver disease. You can see the rates of liver disease in England a bit further down here. And the reason why the Scottish rates of liver disease are so high and they're increasing are because of the terrible diet that most of our patients have and the increase in obesity-induced liver disease, or MASLD. You can see a Scottish delicacy here, which is a battered deep-fried Mars bar, which is quite distinct from the Mediterranean lunch that we had today. And of course, the rates of alcohol consumption, although most of our patients don't tend to drink these expensive single malts that you see down at the bottom. So I'm interested in fibrosis, and the reason for that is, as clinicians, one of our key questions when we see patients with liver disease is how much fibrosis they have. And that's because we know, irrespective of the etiology of liver disease, that the amount of fibrosis in the liver is the best predictor of adverse outcome. So patients who have more fibrosis are more likely to die, they're more likely to develop liver-related complications, and they're more likely to develop liver cancer. We also know that patients who respond to therapy and their fibrosis gets better actually have a better outcome. A lot of the MASLD drug trials have been a bit disappointing in terms of fibrosis regression, but actually if you look at the subgroup analysis and you identify the patients that actually do show evidence of fibrosis regression, they have a better outcome. So more fibrosis is bad, improving fibrosis is good. So because of that, there's obviously a huge amount of interest in developing therapies for fibrosis, so antifibrotic therapies. But as yet, we don't really have anything effective which we can use in the clinic. So why is that? So, I went one slide too far. So when I'm thinking about this, I often kind of relate what we do to other chronic inflammatory conditions. And the best example here is cancer, I think, where there's been huge advances in terms of therapeutics for cancer. And that's, I think, because we now understand the biology of cancers much better. We understand what's happening to the tumor cells, what mutations they develop, and we understand the niche of these tumor cells, which immune cells are present, which vascular cells are present, and therefore how can we use immunotherapy, for example, to try and modulate this niche to try and improve cancer therapy. In the context of fibrosis, we are way behind in this. We still lack clarity on what the phenotypes of the pathogenic cells are, where they're located, how they interact with each other, and even more basic, which patients are likely to progress and which are not. Are there differences in the type of fibrosis that different people have? And, it's not surprising because fibrosis is complex. If you look at a fibrotic human liver, so this is, an example here, actually it's much like a tumor microenvironment. You've got inflammatory cells, you've got adaptive immune cells, you've got vascular cells, you've got obviously myofibroblasts. So it's only really by kind of understanding, unpicking the kind of molecular pathways that are going on in these areas of fibrosis can we really hope to develop targeted therapeutics. Now, I'm going to change this slide in the future and put on ducts in here, which are not in. But so I've done a more generic thing about epithelial cells. So generally, in the context of fibrosis, what we have is an epithelial injury. So hepatocytes generally in Masold or biliary cells in ductal in biliary diseases. And that causes cellular damage. And then it sets off this cascade, which is a complex cascade of different cell types interacting with each other. So you have activation of your myofibroblasts, which produce the scar. You've got changes in the vasculature. You've got adaptive immunity. And for today, we'll talk about the innate immunity or the macrophages, which are key regulators of this process. And we've known for a long time that if you look at areas of fibrosis, whether it's in mouse models or in human disease, actually, yes, you find myofibroblasts, but also these areas are really stuffed full of macrophages. So these macrophages, you can actually identify where the scar is without even doing a collagen stain just by looking at the macrophages. So because of this, there's been a lot of interest over the years in terms of trying to dissect or unpick which types of macrophage are regulating this fibrotic process, which types of macrophage are in this niche. And this, to some extent, has been ironed out in mouse. So I've simplified this, but broadly in the mouse, in the healthy mouse liver, there are 3 different types of macrophage. There are the resident Kupffer cells, and these are embryologically derived, and they self-renew throughout adult life, so without really any contribution from the circulation. There's a small population of macrophages which reside in the capsule of the liver called capsular macrophages. We're not really sure what role they have in the context of human disease. And then there's another population of monocyte-derived macrophages. So in the context of fibrosis in the mouse, what happens is you get a reduction or loss of the resident Kupffer cells, and you get a huge expansion of these monocyte-derived macrophages. And Adrian talked a bit about this, and these are likely to be important in regulating disease pathogenesis. And work we did, this is work I did during my PhD, but also Frank Taka's done a lot of this type of work where we have this model in mice where we have started to unpick what these monocyte-derived macrophages are doing in the context of fibrosis. So what we think is happening is that in the context of injury, you get recruitment of monocytes into the liver. These start off being these pro-fibrotic or pro-inflammatory Ly6C high macrophages. They make multiple inflammatory mediators and fibrogenic signals, which then promote activation of hepatic stellate cells to deposit ECM. And these macrophages are also plastic, and they can upregulate matrix-degrading enzymes such as MMPs or cathepsins, and they can promote the degradation of scar. So we kind of have unpicked this reasonably well in mouse models. What had been much less clear is what was going on in humans. So what are the macrophage subpopulations in a healthy human liver, and how do these change in the context of disease? So the thing that really transformed this is the ability to do single-cell RNA sequencing. And what this meant was rather than us having to know the particular markers of the macrophages in the human liver and then isolate them and do bulk sequencing, we could actually just allow the data to tell us what the macrophages were. So we started doing this back, about 2016, 2017, so quite early days of doing single-cell. And we did, we were able to get cells from either healthy or these are cirrhotic human livers, isolate the cells and do single cell sequencing. So this is all published, so I'm not going to go into the old data, but essentially what we were able to do was to identify a particular population of macrophages, which we termed scar-associated macrophages. And these had a distinct marker profile. So two of the markers I've shown here are TREM2 and CD9. And when we stain human liver tissue, we find that these TREM2 positive, CD9 positive macrophages really localize within areas of fibrosis. They increase massively in the context of disease. And the beauty of having the single cell data is that we can have markers that allow us to isolate these cells. So if we isolate TREM2 positive, CD9 positive macrophages from fibrotic human liver, and we culture them with hepatic stellate cells, what we find is these macrophages promote stellate cell activation. Now, other people have done this in other organs, so we did liver, but there's lots of studies where people have done single cell and kind of similar types of analyses in other fibrotic diseases, so lung, kidney, heart, gut, lots of different studies now. And actually one of the striking things is that the macrophage population that expands in these different fibrotic contexts is very similar. They have a very similar marker profile. They all express TREM2, they all express CD9, they all express SPP1. So we thought, brilliant, we've cracked this. We now know that actually this is the model in human. So monocytes come in, they differentiate into these TREM2 positive macrophages, they start signaling to the ECM producing myofibroblasts, and you set up these circuits where these macrophages and fibroblasts are signaling to each other and propagating fibrosis. So what we thought is brilliant, we need to now think about how we modulate this particular process. Now, as always with macrophages, it's never quite as straightforward as it first seems. So us and others, so particularly Tom Wynn's group, who is now at Pfizer, showed that these macrophages actually promoted fibrosis. So if you isolate these macrophages and you do cultures with stellate cells, etc., they tend to promote stellate cell activation, promote fibrosis. But actually now, because of the interest in this field, there's actually been a lot of other studies which have shown the opposite. So they've shown that these TREM2 positive macrophages actually are anti-fibrotic, so they promote fibrosis regression and inhibit fibrogenesis. So what we started to wonder was, well, could there be more heterogeneity within the scar-associated macrophage compartment than we first recognized? Now, what you need to realize here is that single cell analysis is a numbers game, okay? So the more cells you have from the more patients, the more chance you have of detecting subtle differences or rare subpopulations, okay? So this work was done by Kaishin in my lab. So what we decided to do was we decided to atlas all of the published human single cell RNA sequencing data, okay? So that's actually from 8 studies, but only 7 of the studies actually made it through the initial QC. So we went back to the raw data, we downloaded the raw data, we redid all of the analysis, redid all of the clustering, redid everything from the beginning. And we end up with a data set from 77 patients, 42 of them who had a healthy liver and 35 who had some form of CLD. Now, I don't expect you to read this, but just to show that the data set is very complicated. So what you end up with, if you do the final annotation of all of the different cell types that are captured, is 125 different cell states. And we were very careful not to overcluster it. So these all have biologically plausible marker genes, okay, of the different cell types. So that's what we call the level 3 annotation. There's 125 different cell states. Clearly, that's quite a difficult data set to work with. So we've kind of made a level 2 and a level 1, which kind of groups transcriptionally similar cells together. Just depending on what you're using the data for, you may want to only sort of have hepatocytes grouped together rather than different subpopulations of hepatocytes. And just to show what the benefit of this kind of large-scale data, if we go to our level 2 atlas, which has 34 cell types, what we can start to do is identify rare populations. So things which were not identified on the original studies, things like basophils, mast cells, mesothelial cells, neurons even, we can identify from this kind of larger scale atlas, and we can now start to unpick marker genes which differentiate these different cell types. So for the purposes of today, I'm going to focus on the macrophages. So what we then did is we looked at the level 3 atlas of the macrophages, or mononuclear phagocytes, and we find 14 different cell states here. And if I draw your attention to these, we find actually there are 2 different types of scar-associated macrophage. So both of these macrophages express the markers we previously defined, so TREM2, CD9, etc. So we wouldn't have been able to pull these apart in the original study, but actually now with more data, they're partitioning into 2 separate populations. We can show, well, how do they change in disease? So both of these populations expand in the context of CLD. So you can see particularly scar-associated macrophage 2, but also scar-associated macrophage 1. I'll not go into this today, but we've done a very similar analysis for all of the other cell types. So there's good vascular cell data, Jordi, if you're interested in looking at it. So, and Cation's made a public web browser, and I'm happy to share the data if anybody's interested in looking on their cell types of interest. So if we go back to the macrophages, so what we wanted to know is, what are the differences between these two different types of scar-associated macrophage? So if we do a differential expression analysis just between these two populations, we find there's a lot of differentially expressed genes between the two. If I draw your attention to on the right here, the particular interest was in inflammatory and fibrogenic mediators. So what we could see is that scar-associated macrophage 2 was the population that was enriched for traditional pro-inflammatory fibrogenic mediators, so IL-1 beta, TNF-alpha, PDGFB, TGF-beta1, etc. Whereas the scar-associated macrophage 1 population was actually enriched for more anti-inflammatory, pro-resolving mediators, things like IL-10, MMPs, etc. We also wanted to then look, well, are there particular receptors that allow us to distinguish these different populations? And what I'll talk a bit more about this particular receptor, OLR1, which we found to be enriched on the more pro-inflammatory, pro-fibrogenic phenotype of scar-associated macrophage. The other thing that made this interesting was the fact that when we look at our atlas, OLR1 is only expressed by these cells in the liver. It's not expressed by any other cell type, so allowing kind of validation or therapeutic manipulation to be, more accessible and tractable because we're not unlikely to have any off-target effects. So if we do immunostaining, we can find that these OLR1 positive macrophages are localized within areas of fibrosis in the human liver. And they're different to the GPNMB positive macrophages. So I showed GPNMB was a marker of the scar-associated macrophage 1. So the OLR1 and GPNMB positive populations are different. So there are two different types of scar-associated macrophage in the human liver. Now, one of the challenges with a lot of the single cell data is that it's really biased towards patients with end-stage disease. So mostly these data sets are generated from patients at the time of liver transplantation. So what we wanted to know is, well, are the genes we identify on these macrophages, are they relevant in earlier stage disease at a stage when you might actually think about an antifibrotic therapy? So what we did was we went to the steatocyte dataset, which is a dataset generated by my colleagues in Edinburgh, Jonathan Fallowfield and Tim Kendall. So what they had was this dataset from 940 patients with MASLD from around Scotland, and they had liver biopsy tissue and histology and electronic health records. And on 700 of those patients, or 707, they also had bulk RNA sequencing from the liver biopsy tissue. So we could then look at OLR1 expression, and what we see is that OLR1 goes up in these biopsies with fibrosis stage. So this is not just a feature of end-stage disease, this goes up alongside fibrosis in earlier stage disease as well. Now, the other great thing that they have with the steatocyte data is clinical outcomes, so allowing us to look at expression of our particular genes of interest and determine whether they're linked to clinical outcomes. So when we look at OLR1 expression, what we see is patients who have high levels of OLR1 expression on their baseline liver biopsy are more likely to have liver disease progression and a liver-related event, and they're more likely to die. So you can see that's pretty striking on the left and the right there. Now, we've done very similar experiments in mouse. So this is the CCL4 model, where we've done single cell sequencing, and you can find these two populations of scar-associated macrophages, and this was done by Eleni, who's a former PhD student in the lab. And when we look at the similar kind of differential expression, actually we see a very similar change. You have these OLR1 positive macrophages that are more pro-inflammatory. We can also start to do more validation experiments, so we can prove at a protein level that these macrophages make more pro-inflammatory cytokines, so more IL-1 beta, for example, here. The beauty of the mouse model is we can do some things we can't do in humans, so we can look at kinetics. So if we look at flow cytometry of these different macrophage populations, what we find is that both types of scar-associated macrophage are really in the liver at the time where you've got active injury and fibrogenesis. But when the injury is resolved and regressed, actually these macrophages are largely gone. The other thing we can do in the mouse model, which is difficult to do in human, is fate mapping. So one of the questions that's always asked when you find different macrophage populations is, are they coming from these resident Kupffer cells or are they coming from monocytes? So to try and address this, we did this experiment, which is a bone marrow chimera experiment. But rather than using irradiation, what we did was we gave the mice a chemotherapeutic agent, busulfan, and then we transplant the CD45.2 mice with CD45.1 positive bone marrow, allow them to reconstitute, and then we can introduce chronic liver injury. Now, the beauty of this as opposed to irradiation is if you look at the resident Kupffer cells, they're not replaced by the bone marrow-derived cells. So they remain of recipient origin, whereas the monocytes are replaced, or at least partially. So then when we do injury, we can then look at, well, where did these cells come from? Did they come from the blood, or are they more likely to have come from the liver or from the resident Kupffer cells? So the resident Kupffer cells remain largely of recipient origin, but both populations of scar-associated macrophages are more like the blood cells, so proving that these cells are coming from the blood rather than the resident Kupffer cells. So are these OLR1 macrophages functional? So we want to go back to the human system. So what we did was, we've done a few different things, but what we did in this experiment was we tried to make some spheroids. And these were ES-derived spheroids from human cells, where the ES cells were differentiated either into stellate cells, into hepatocytes, or into endothelial cells. And these are then aggregated into a spheroid. And to this, we added primary monocytes from patients with liver disease. And you can then mature these for 8 days, and then actually you can potentially do drug treatments from like day 8 to 15. And when we then stain these spheroids, what we find is that you find IBA1 positive macrophages are embedded within the body of the spheroid, really showing that there's been engraftment of these monocytes into the spheroids. So then when we look at gene expression, what we find is that these macrophages have developed this kind of OLR1 positive scar-associated macrophage phenotype. So you get an upregulation of OLR1 and other scar-associated macrophage markers, and you also get a spike in fibrogenesis. So you can see COL1A1 goes up, and there's a trend to increase in COL3A1. On the other side of that, if we deplete these macrophages during the treatment phase, so this is with a CSF1R inhibitor, we get a reduction in the scar-associated macrophage markers and the pro-inflammatory cytokine production, and we get a reduction in fibrogenesis, showing that these monocyte-derived macrophages in human samples and human models are actually promoting fibrosis. So I've talked a lot about OLR1, but what is OLR1? Well, there's actually very little data in the liver on OLR1, so it's best described in the context of atherosclerosis, where it's reported to be it's an oxidized LDL receptor expressed on endothelial cells in atherosclerosis, but also in macrophages, where it's thought to promote pro-inflammatory signaling in that context. The other question is, what are the ligands? Oxidized LDLs or lipoproteins probably do have a role in liver disease, particularly in MASLD, but actually there are lots of other potential ligands which might also have relevance. So that's something we're working on at the minute, is trying to figure out, what are the potential ligands in the context of human liver disease. So is it functional? So if you go back to human cells, so either THP1 macrophages or primary human monocyte-derived macrophages, and we block or attenuate OLR1 expression, so this is an siRNA experiment done by Fabio, who's a postdoc in my lab, you abrogate inflammatory cytokine production, so suggesting that OLR1 signaling in macrophages is promoting inflammation and fibrogenesis. And if we then kind of make the model slightly more complex, where we take the primary macrophages and we either knock down OLR1 with siRNA on the left, or we block it with a blocking antibody on the right, and we then take the conditioned media from those macrophage modulated macrophages and look at what it does to stellate cells, we find that when we blocked or knocked down OLR1 in the macrophages, we're abrogating fibrogenic activation of the stellate cells, so suggesting that OLR1 signaling in macrophages is promoting a fibrogenic phenotype in stellate cells. So based on this, I think we are having to refine our model of what's going on in the fibrotic niche. And actually, there are two different types of scar-associated macrophage. So there's a more pro-inflammatory scar-associated macrophage, which has these pro-inflammatory cell circuits with the activated fibroblasts, and that's likely propagating injury. And there's a more anti-inflammatory stroke pro-resolution scar-associated macrophage population, which has different interactions with the stellate cells. So currently, I think targeting this population is more likely to be tractable as an antifibrotic approach. How am I doing for time? Okay. Hopefully I've shown you that having this large-scale single-cell data from multiple patients actually is a useful resource to allow us to really have a better handle on disease-associated cell types and to better define therapeutic targets. And I've shown you an example here of how we've done this for the macrophages, but of course you could do a very similar type of analysis for other cell types to really try and unpick some of the complexities of the interactions between these cells in fibrosis. So, in the last kind of few minutes, I'm going to talk a bit more about the spatial side of things. So what about the spatial organization of these cell types in fibrosis? So I've shown this already, but fibrosis is a very spatially patterned process. So, we know that there are very distinct cells and interactions within these areas of fibrosis compared to more healthy or functional areas of liver tissue. So what we really need to know is which cells are active and functioning in this particular niche. And in the single cell field, this is the kind of an analogy that's often drawn where, the bulk sequencing is like a smoothie. So, you can tell broadly the differences between different groups. We've kind of partitioned the smoothie into its constituent parts with the single cell. So we know which fruit is actually in the smoothie. But what we now really need to know is how that fruit is organized, how these cell types are organized within these areas of fibrosis. Now, we've heard it already mentioned briefly, but this has become quite a complicated area, spatial omics, and there's lots of different platforms, lots of different methodologies, and this is something that's fast evolving. Broadly, I like to think of it in two categories. So there's the more sequencing-based approaches, and you'll have seen things like Visium or StereoSeq or Geomics, which are more sequencing-based. I'm not going to talk about any sequencing-based data today. I'm going to talk about the more imaging-based approaches. So there's high-plex in situ hybridization, and there's a variety of platforms for this, Cosmic, Xenium, etc. And there's high-dimensional antibody staining, and again, various platforms for this. So I'm just going to show you briefly a couple of examples of this in the liver. So for Cosmic, so Cosmics is a nanostring or now Bruker platform. So this is a high-plex in situ platform, and what you do is you take your tissue and you put it onto a slide. It's FFPE tissue. You permeabilize it, and then you're essentially staining that with a 6,000 plex RNA in situ probe set. Okay, so theoretically it can pick up 6,000 different RNAs or different genes. And then you image it, and all of that's done in a fairly automated manner. You can't really control any of the parameters or anything. So just as a technical tip, these are very expensive experiments, but it is possible to put multiple livers from multiple patients onto each slide to make it more cost effective. So, you can see we get kind of 6, 8 or bits of liver on each slide, kind of making it more cost effective in terms of data generation. Adrian mentioned this earlier, but segmentation is really crucial to this. So, you can't just trust the segmentation you get from the standard workflow. So this work was done by Jackie, who's a PhD student in the lab. So if you take the standard outputs, you can see there's lots of kind of gaps in the segmentation and lots of errors and that. So Jackie worked very hard in trying to optimize this to try and get a better segmentation. And you can see we've, I think, done a really reasonably good job, but Adrienne may have other comments on that as a segmentation expert. But I think it's reasonably good. Maybe not perfect, but it's better than it was. So we've done, in overall, about 20 different samples of liver here. And one thing I want to draw your attention to is, well, two things on this slide. One is it's reasonably consistent, which is good. So even different patients fixed for different amounts of time, actually the amount of gene you detect per cell are reasonably consistent, but it's clearly not 6,000. So if you look at the mean number of genes you detect per cell, it's kind of the order of 300, 400, 500 genes per cell. Okay, so you still have a lot of dropout and a lot of sparsity in this type of data. However, you can start to get biologically meaningful results from it. So we can annotate this. So this is the data from the Cosmics, which has been viewed like single cell data. So we can annotate the different cell types. This is kind of a top level annotation, but yes, we can do more fine grained annotation, but using our single cell reference to kind of help guide this. And even this across multiple patients is pretty consistent. You don't see a lot of batch effect in these across the different patients, even when we put data from 20 different patients together. And you can get some nice images. So this is just an example. So we're still working on the analysis, but essentially we start off with this antibody stained bit of liver tissue. So those are the 4 antibodies on DAPI. We then run our segmentation. So you make these cell boundaries from the segmentation. And then each cell has a transcriptional profile, which we've annotated. And then you can map the annotation back onto the tissue. So this is this level 1 annotation. So very top level, what we can find is, the hepatocytes in the nodules, not surprising, mesenchymal cells in areas of scar. But if you can start to do the more fine-grained annotation, you start to get more patterns of different cell types and where they reside. Okay, so if I draw your attention to the macrophages, so what we can start to do is, the 2 populations of scar-associated macrophages which we identified on the single cell data, we can annotate in this particular data set, and we can now look, well, where are those macrophages located in the tissue? And we can take it a step further. Well, what cells are they neighboring? So, for example, the activated stellate cells you can see are in the same niche as these scar-associated macrophages. And these are different there are different fibroblast populations which are more in the middle of the scar with actually, interestingly, a B/plasma cell signature, which tends to be more in the middle of the scar. So really allowing us to start to get the spatial patterning of areas of fibrosis. And we could take this even one step further where rather than analyzing the cells, we can analyze this as neighborhoods. So for this, we take each cell and we look at, well, what are the neighbors of that particular cell? And then we run a clustering algorithm on the neighborhoods, and you can kind of get these different niches, which are niches of cells which are located in like that same pattern across the different bits of tissue. And we can start to pull apart, actually, there are probably different types of scar which have different cellular compositions and forming these different niches. And we can go back and say, well, which cell types are in these different niches? And, this is still very much a work in progress, but for example, we find these niches at the bottom here which are enriched for macrophages and pro-fibrogenic cells, and a different niche up at the top here which is more an adaptive immune phenotype. I think I'm running out of time, so I'll not go into this too more, but you can do a very similar thing with protein data. So this is high-plex antibody staining. So, and these antibodies are bar-coded. So rather than the antibody being stripped in Adrian's model here, the reporter stripped, so you image it cyclically, but the antibody stays on the tissue. And we've got this kind of panel now, which takes a lot of optimization, but it's of about 65 different antibodies labeling all of the different cell types in the liver. And we can do a very similar type of clustering analysis that we do for single cell, but this is more on protein, and identify the different cell types in the liver. And we can start to do similar types of analyses that I just showed you for the cosmics, where we segment the liver, and because we now have protein data, we can map particular cell types to different niches. So we'll end up with, I think, gene expression and protein data showing us these different niches and which cell types are in them. And I think this protein data is particularly useful for things like T cells, where it's difficult to annotate them using just transcript alone. So this is still in its infancy, I would say, but it's a fast-moving field where I think spatial omics is really going to refine our understanding of which cells are in the fibrotic niche, the molecules, and how they're interacting with each other. And I think we're already starting to see that actually there are probably different types of fibrotic niche which have different cellular compositions. But what we don't yet know is what are the functions of these different niches, how do they evolve in different types of liver disease? Is one present earlier in disease than others? So I think as we go forward and generate more data, we'll hopefully get a much better handle on that. So obviously, lots of people to thank, particularly the people in the lab who did the work. So I've mentioned them as I've gone along. Jackie, in particular, who did a lot of the spatial work, and the analysis of that is still pretty tricky. And Jonathan and Tim, who we work with on the steatocyte data. So thank you very much. I'm happy to take questions. Obviously, also happy if people want to email me, if they were interested in the data or anything, then feel free to email me. I'm happy to collaborate or whatever, no problem. So thank you very much. We have not been able to do that yet, and there's a couple of challenges in that. One is, as I mentioned, that most of the single cell and spatial data as it is currently is from end stage cirrhosis. So I think a lot of the interesting different niches are going to be earlier stage disease when you're looking at needle biopsies. And we've started to do a bit of that, but we've not yet invested the time and the money doing all of the spatial omics on needle biopsies, but I think that's something that will come. And the beauty of the spatial is that it can be done on FFP sections, so you can go to your biorepository and you can run spatial on whatever diseases you like. The problem is the money. So currently it's very expensive. So but in the future, I think in 5 years' time, we will have a much better handle on that because we will have done these analyses on different etiologies, different disease stages, and have a much better idea of what's going on. Yeah, absolutely possible. I think statins, actually beta blockers, probably modulate the macrophage phenotype to some degree. So actually a lot of stuff we use probably are potentially modulating the macrophage phenotype. And statins are interesting in this context because, as you say, oxidized lipids might be reduced with statins, which might attenuate and pro-inflammatory macrophage function. Actually, it's more directly druggable. So there's a monoclonal antibody which has been given to patients who've got atherosclerosis. So actually, I think you could probably block it directly actually to figure out whether it's having a direct effect, because I think the thing with statins is it's quite complex to unpick what the mechanism is. Yeah. So both difficult to answer. So the first point about if the location of the injury might regulate the scar-associated macrophages. So that's difficult to answer in humans currently. Kind of relating to the point that we were talking about before, where a lot of the analysis is on patients with pretty advanced disease, so they've got portal bridging already. Yeah, so the macrophages are in those areas. So actually, I think you would have to do more analysis on biopsy tissue to actually get a handle on whether the pattern of injury is influencing where the macrophages are. In mice, yes. So if you've got CCL4 where you've got pericentral injury or you've got biliary injury, you find the macrophages in the injury niche. So, I would suppose that it might be similar in human, but we don't have any hard data to back that up. If you analyze the human data, these are patients who've got advanced liver disease, actually, you don't really find the same monocyte-derived Kupffer cell that have been reported when you deplete Kupffer cells in a mouse, So there are macrophages that maybe have slightly more of a Kupffer cell phenotype, which are different to these scar-associated macrophages, but it's different in the human, So, and in mice, if you wipe out the Kupffer cells, so if you do collect for FDTR and you wipe them out, monocytes will come in and adopt that phenotype. But in the context of a more kind of standard injury model, those monocyte-derived Kupffer cells are not in the niche of, we're finding fibrosis. So I think the monocyte-derived Kupffer cells are probably not the cells that are regulating the injury and fibrogenic response because they're not in the same niche. But it's probably model dependent, to be honest. So that's why it's been a bit confusing and a bit tricky to unpick that. I think the edge of the scar is more fibrogenic. Okay, so there's more active scar deposition around the edges. So that's where you see the macrophage. That's where you see ducts, actually. That's where you see the ductal reaction. So I think that's where the active scar deposition is happening. And in the middle, that's probably old scar. So that's scar that's been there for years, decades, whatever, in these patients. So I think if you're thinking about a therapeutic, actually you want to be focusing on the actively fibrogenic areas, so the areas around the edge of the scar. Still need more data to nail down that point, and it's challenging because mouse is different, so you don't get that same thing in mouse. In mouse, it's all actively fibrogenic, so doing it in any form of model is quite difficult. So I think we're largely reliant on the observations from human disease to kind of get at that point. And part of that is practicality. Actually, as I've shown you, hopefully, that I think the best way of looking at this type of thing is single cell analysis, because it's allowing you the data to tell you what the differences are. And there have been technical challenges doing single cell on biopsies from intermediate stage disease. So what lots of people do is single nuclear sequencing, which gives you decent data, but actually the macrophage data on that is not great. So we have a protocol now which works, but it's taken a lot of time to optimize, where we can get decent single cell data from a needle biopsy and build up a data set of, well, what are the features of the macrophages at a time in disease when actually you might intervene or might modulate them. Provisionally, actually, they're quite similar. So I don't think this is just a feature of advanced liver disease. And provisionally, it's not just MASLD. You see the same types of macrophage in different types of liver disease. So I think this is a common injury phenomenon. But I think, we need to work more on fleshing that type of data out to try and properly understand, etiological specific and disease stage specific features of these macrophages. So I think we have to also be careful with the nomenclature. So there's, I think, fibrotic and fibrogenic. So because fibrotic is, I guess, where you have collagen. So within that, I think there are areas which are more fibrogenic, so actually active expression of collagen. And absolutely, yes, you can use that niche analysis to look at, well, which are the niches that have more active fibrogenesis, more active collagen production, and what are the cells contained within that niche. So you can do that totally, and that's something we will do. It's just the initial stages of the analysis have taken a bit of time, but we will try and answer that question. But what the provisional data would suggest is that it's the areas where the macrophages are and the activated myofibroblasts, perhaps not surprisingly, and actually activated ducts in those kind of edges where you've got more active scar production. So I think that's probably the hot spot for fibrogenesis. I'm certain that the composition of the extracellular matrix will influence the phenotype of the macrophage. Years ago when I was doing my PhD, we did some in vitro experiments where if you put the macrophages on different extracellular matrix components, it affects the phenotype. So I suspect absolutely the local milieu of the extracellular matrix will influence the macrophages and the other cell types. We haven't got at that yet. I think you probably need some form of spatial proteomics to really allow you to get at that, which, I think will come. It's just not quite there from a technological point of view yet. So we've got you identify most other immune cells. Neutrophils are difficult, mainly. So this is single cell data, and neutrophils do not like single cell. So if you do a 10x experiment on neutrophils, essentially the neutrophils generally have died before they've even gone near the 10x. So you don't get great data on neutrophils. So we can identify the neutrophils, but I'm certain they're underrepresented in the data set. So I think we don't really interpret too much on the neutrophils currently. DCs, so the DCs, where they're there, we can identify them. They don't really seem to change a lot in disease, actually. If you look at either at number or transcriptional profile, there's not a big difference in the dendritic cell compartment. So the things in the mononuclear phagocytes that change most are the macrophages. So not excluding them having a role, but we focused on the things that actually seem to change most in disease.
So thank you very much to all of you for your participation, especially Adrian and Prakash for their beautiful talks. I'll remind you that the liver seminars continue tomorrow. So we have two great speakers coming. So Jonis Sovo from Harvard and Donato Inverso from Milan. So please, I hope to see you all tomorrow for the next session of the liver seminars. Thank you very much.